The opinions expressed by the host and guests in this show are not necessarily the views of Children's Health Defense.
Good morning, CHD. It is Friday, January 23rd, 2026, and I am Kim Mack Rosenberg, General Counsel of Children's Health Defense. We have a great show for you today. Rick Jaffe is joining us, and we're going to be discussing CHD and other plaintiffs' new lawsuit just filed this Wednesday against the American Academy of Pediatrics. And after that, Brian Hooker with Doctors and Scientists. His guests today are Jessica Rose and Kevin McKernan, and they're going to be discussing COVID vaccine contamination. Rick, thank you so much for joining us today.
Well, thank you for having me.
I know you were on yesterday talking about this briefly, so for people who saw it yesterday, they may hear a little bit of the same information, but also there's so much more. We have a little bit more time. We can delve deeper into this. Can you give people sort of an overview of what the lawsuit is about and what RICO is? Because not everyone understands what a RICO claim is, or they may think it's, they've heard it in connection with, for example, mobsters. So can you explain that to our viewers?
Well, RICO is a federal statute that's been around for decades. Originally, it was enacted to go after the mafia. But since that time, many lawyers have tried to use it to go after business entities, their competitors and whatnot. So the reality is RICO as a statute has gotten kind of a bad rap because it's been overused by business litigators. But the point is that it's just a statute, right? And a statute, it's a civil statute and it's a criminal statute. And the statute has elements. So what a lawyer has to do is take a look at all the elements of the RICO statute and see whether you have facts to support them. And that gets to maybe the most important part of our case, which is that our RICO case is modeled on the RICO case filed by the US government and many insurance companies against the tobacco industry for their fraudulent statements that that smoking was not connected or not proven connected to cancer. So for decades, that was what the tobacco industry was saying. And what they had is they created these at least two organizations, the Tobacco Institute and the Center for Tobacco Research, which were nominally research institutions, independent research institutions that found that dismissed all the studies that showed a connection between smoking and cancer. And they created a false sense of lack of proof, right? And they said the science is unsettled. And after many years of litigation and different forums with insurance companies and the US government, it was held in a lot of these cases that scheme using science, so-called science, to show a lack of proof as to the connection between smoking and cancer was a RICO violation. It was a fraudulent scheme. And where we filed the case in the District of Columbia, that's where the principal case was litigated, the US versus Philip Morris. And it went up to the Court of Appeals, and the Court of Appeals affirmed this like 1,000+ page decision after a trial that the tobacco enterprise, consisting of the manufacturers and these organizations, committed a fraud and RICO violation. So our case is directly based on and patterned after the tobacco industry litigation. And we have a section in the lawsuit which just shows the similarities. So the big hurdle that we have to get over is this idea that RICO is just about mobsters. It's just a statute, and you have elements. And if you have knowingly false misrepresentations of facts submitted through the mails or the wires, the internet, then those are predicate acts of racketeering. And if you have two of them, and if you have an enterprise, and you have proximate cause, and then you have injury, that is RICO. One of the other things that the novelty in this case, sort of, is that the way the AAP works is that the delivery system of the misinformation is via its 67,000 member pediatricians, who essentially are the interface with the families in America. And that's how the fraudulent statements were communicated, in addition to AAP's website and journals and all that. And that's the other thing that people have to realize: when the instrumentalities of the fraud are delivered via intended parties like the pediatricians, that is proximate cause. So I think the bottom line is, in a sense, it's a novel action, but it's directly predicated on principles that have been affirmed through the tobacco industry. Really, the only difference is people kind of viewed the tobacco industry badly at the time, where they don't view the AAP badly now, because frankly, they have better PR. So that's essentially the underpinnings of our lawsuit. And here is the basic fact of our case, right? For the last 25 plus years, the AAP has been telling families via its members, the pediatricians, that the vaccine schedule has been fully tested and proven safe. That is false. The vaccine schedule has never been proven to be safe. It's not Rick Jaffe saying this or my lawsuit saying it. It's the Institute of Medicine saying it in 2002. All right. And they asked for the studies to be done. And years later, in 2013, they checked back and the studies still had not been done. And they again asked that the studies be done to show that the cumulative effect of the vaccine schedule was safe. And those studies have never been done to this day. And here's the RICO part of it. The RICO part of it is the reason the studies have never been done, or a large part of the reason, is that the AAP and its enterprise associates have been blocking the studies, right? They've been blocking the studies that would show safety. And all the studies that were done, like by Paul Thomas and the recent study by Hooker and Carl, which we'll talk about, they've been discredited. And that's part of the practice. You block the studies that you can block, and then you disparage the studies that you can't block. And that fits the RICO statute.
And I think one of the things, I think that the tobacco model is a very easy one for people to understand, even lay people who are going to read the complaint or who are listening to us have a discussion. I think that's an easy one for them to follow, but I think AAP and the other enterprises go even a step further, right? The tobacco industry is saying the science is unsettled. Here, they're saying the science is settled, that the science has, as you said, that the studies have been done where they have not been done. And then there's a section of the complaint where you talk about this idea, the phrase we often use is creating customers for life.
Right.
That, the tobacco industry was doing a number of things that were wrong and obviously fell within the RICO statute, but they weren't running oncology clinics, as you point out in the complaint,
Right.
when people are developing cancer from smoking.
Right. That's right. And I got to make one correction when you talk about what I say in the complaint. Let me just point out, it's not just me; it's you and Mary Holland.
No, trying to give you credit here, Rick.
Right. Thank you. I appreciate that. But actually, there's a specific point on the racket, that was actually both of you were married. So that was your guys' major contribution, but that is absolutely 100% correct. The enterprise's actions in this case is much worse than tobacco, because what they have done is they have financialized the conditions that they create. So they create all these side effects. And again, this isn't Rick Jaffe talking, right? We know that vaccines have side effects because they are listed in the package inserts approved by the FDA. So what we've done in the complaint, and this is the seed of this was Mary and your idea, what we've done is we've lined up the side effects that these vaccines have, and then we showed how some of the vaccine manufacturers bought the companies that treat the side effects. So it's like, you know what it's like? It's like the tire repair guy throwing nails in the street, and then his business booms. So they're literally giving drugs that create the side effects, and then they buy the companies that treat the side effects. I mean, I have to say, it's a pretty good business model. It's not for the American kids, but it's an understandable business model.
Right. They're either buying these companies or they're developing the products themselves in-house to treat these conditions. Now, let's talk about the plaintiffs here, particularly the parents, the families who are plaintiffs here. I think that Dr. Thomas and Dr. Stoller, we can talk about them as well. They're well known to our audience. But tell us about the families who are plaintiffs.
Well, all of them are bad. All the stories are bad, but the worst, the thing, and why I put the Andrea Shaw as the lead plaintiff, that's the worst story I've ever heard, but it's par for the course. Basically, these twins were vaccinated despite the concerns of the family about a past history of vaccine reactions, but they were told that that's no problem. And then the twins died like 8 or 9 days later. When they went to the hospital the day after, it actually wrote down it was the condition was a vaccine injury, and yet the mother's being investigated for homicide, which is just horrendous. I mean, it shows you how the mindset has just infected society, where you have a straight shot from vaccines and a reaction, which is documented in the medical records, and all of a sudden the parents become homicide suspects. Too much heat, postpartum depression, all kinds of crazy theories. And I think that is really, you can trace a direct line to that from what I call the fundamental fraud, which is this notion created by AAP in around 2001 or 2 that children could handle 10,000 vaccines. And that was I call that the fundamental fraud, because what had happened is back in the late 90s and the early 2000s, when the vaccine schedule increased, not as dramatically as subsequently, but it increased, and parents were concerned about the safety of administering all these vaccines to their children, because it's just not the antigen. You have all these adjuvants in there, and then, you have polysorbate, and you have a lot of stuff that's in there, which ultimately hasn't been really tested, and they were concerned about that. So what happened is, in response to those questions, AP put out this article, which in effect was a sleight of hand. People were concerned about the toxicity of the vaccines, and what this article really argued, and it's in the complaint, and it's cited, and the title of the article is something like "Why Parents Shouldn't Be Worried About All These Vaccines." What they did is they gave an answer about how many vaccines a child could respond to at one time. So it wasn't really a toxicological answer. It was an answer about immunogenicity, whatever it is. So talking about the response, the number of responses, how many vaccines could be responded, it was a completely different answer. It wasn't an answer to the question that was asked. And for the last 25 years, that's been the reason, I think, why pediatricians take the position that you can do as many vaccines as necessary on the catch-up schedule because of this misdirection, that the child's body could handle 10,000 vaccines. And I think that's, like I say, I call it the fundamental fraud. I think that's I would go so far as in healthcare. I think that's been one of the most damaging pieces of information out there, maybe ever, because it's justified injecting kids with multiple vaccines at the same time. And the irony is, and this is, and all this stuff, took us months, the three of us months to put this all together. The irony is that, after the Gulf War, the Brits did some study that showed basically that the more vaccines you gave to the troops, the sicker they became. And the result of that was, and is, as of this year, if people who want to join the military show up for induction or officers candidate school, they say flat out that we'll only give you 5 vaccination for 5 diseases at one time. Otherwise, we're going to disqualify you, because even the military, the Marines, this is the Marines, they understand the danger of multiple vaccines, but these super fit 18 to 22-year-olds, have a limit to the number of vaccines you can get, but 6-month-old infants, 2-year-old infants don't. And that to me is just appalling.
And Rick, there's a plaintiff in this case who I think really demonstrates the impact of that catch-up schedule. Can you talk about that family?
Sure. This is the family of the second plaintiff. I mean, they got, I think it was vaccines for 11 different antigens. And then, I think the child died in the hospital the next day. And the interesting part was that there's information that the child had brain swelling. And this is the other part of the enterprise and how it works. But the death certificate didn't mention that, and just did the sudden unexplained death syndrome, SUDS or whatever it's called. So this is the cover-up. Routinely, as a matter of practice, it's very rare for a death certificate to show death caused by vaccine. But this is a classic case, the Nelson child, Carter. I mean, it was just horrible. I mean, it was less than 24 hours later, and there's evidence that the autopsy, the coroner found the brain swelling and just didn't put it in the death certificate, which I think is quite common. So I mean, that's just it's beyond words that this kind of thing happens in this country.
And the third plaintiff is a Jane Doe. And her daughter is still alive, but her life has been catastrophically altered by vaccines. Can you tell us about that family as well?
Right. And here's a different part of the problem. All right. And this is a case where really the doctors got it right, but the AAP model, this 10,000 vaccine nonsense, compelled a different result. So this child had severe allergic reactions to egg-based vaccines. So the pediatrician wrote an exemption for egg-based vaccines. And by the way, kudos to the pediatrician, because I don't think that is a contraindication or precaution under AAP or ACIP, the Advisory Committee on Immunization Practice. I don't think that's even a valid contraindication. But the pediatrician did it. And then later on, the child received a non-egg-based shot, DPTA, I think, and had a severe reaction to that. And then as a result, the pediatrician gave an exemption for all vaccines because of the reactions to egg and non-egg-based vaccines. And again, kudos to the pediatrician. She got it right. And she, in effect, disregarded the contraindications and precaution guidelines by AAP. And so then what happened is eventually the medical consultant or director of the school rejected the exemptions because they weren't based on AACP, and they wanted a renewed exemption, which the pediatrician gave. And then the director, who, by the way, never saw never treated the patient, never did really anything, wanted a letter from an allergist confirming all of these allergies. And Jane Doe got that for him too. And then, but the medical director / consultant rejected that as well and insisted that the kid get vaccinated. So this was a big problem. And then, so they wouldn't let her be in school. She would show up and then she would have to go home after 3 hours. It was a horrible situation for her. She was deteriorating mentally, and the family decided that they had no choice but to get her vaccinated, and they did. And of course, she had horrible reactions to that. And she suffered from documented side effects of this vaccine, as both treating physicians predicted. So she had surgery and she still has ongoing problems. And what this shows is, like I say, different. It's not the AAP physicians being wrong. It's they got it right, but this medical director looks at this cookbook in the last section of the Red Book, which is the AAP Bible, and they don't see a reason not to give a vaccine because of a minor or an allergic reaction, and decides to overrule 2 treating physicians, basically because it's, the Bible says they can do it. And so that explains another part of the problem, and how the problem is broader than just the pediatricians. It's the mindset of the ancillary people involved, like these school consultants or like this police department, that somehow can't figure out that the vaccines killed these 2 kids. I mean, it's really, when you think about it, it is just hard to believe.
It really is. And we could probably talk about this all day, but we do have to wrap up. I have sent the complaint, as I'm sure you have, to many, many people. I always say to people when I'm sending it, read it, follow the links. You'll see that everything in here is very well documented. This complaint tells a compelling story. Rick, as we wind up, can you tell us what next steps are now that the lawsuit has been filed?
Well, I think a couple of things are going to happen. Eventually, I'm sure the AAP they have to get served. They'll move to dismiss. I have a feeling, let's see what happens. I think we've told a story, all right? We've put together things that haven't been put together before, maybe. So I think all kinds of good things could happen. I think it's possible. It just because of, I don't know, maybe this is the field, but the same day we filed, the Paxton, the Attorney General of Texas, announced that it's sending out civil investigation demands to a lot of the mafia, a lot of the people we say are enterprise associates. So they're doing that. I'm in communication with another state. I in part, we did this. You, me, and Mary, we did this to alert, to tell the story to people, a story that people don't know. And it's kind of a guideline. And, we'll see what kind of reaction we get. I mean, what I'm hoping is going to happen, some people will read it. It'll have information they had never thought before. It might change their minds. Some government people, I think, have been thumping around trying to figure out to get a handle on this. So I'm hoping that our lawsuit is going to help them get a handle, and maybe some government agencies get involved in some way. So I would say what's next is, and then the other thing is, I think when you do file stuff like this, things start coming out of the woodwork. Former AAP people, because I think there's a lot of stuff out there like there was that was uncovered through discovery in the tobacco case, where the fraud was really written down. So I have a feeling that all happened in this case as well. And with a little luck, maybe some of that gets back to us. And by the way, should I mention, thank you very much, CHD, for funding this. I mean, it was a bold move, and I think the community owes the CHD and you, Kim and Mary, a debt of gratitude for that.
Well, and CHD is also a plaintiff here. But Rick, thank you so much for coming on today. Again, like I said, we could talk for hours about this, but stay tuned, folks, for Doctors and Scientists with Brian Hooker, Jessica Rose, and Kevin McKernan talking about COVID vaccine contamination.
Thank you.
Well, good morning, everybody. Happy Friday. It is Friday, January 23rd, 2026. Such a good year. This is going to be such an amazing year. And I want to thank you for joining us. I'm your host, Brian Hooker, for Doctors and Scientists. And I want to bring on our guests right away, because when they talk, like, things happen and people notice. When I talk, well, maybe, yeah, some days on a good day. But here we have Kevin McKernan, CSO of Medicinal Genomics, and Dr. Jessica Rose. You have very recently published a paper. It came out in the Journal of Independent Medicine. How do you do this astounding work? It's like, I think I'll do something groundbreaking today, and then you just go do it. So Kevin, let's start with you. What was it, tell us, I'm kind of keeping it secret because I want you guys to do the reveal and not me. But Kevin, tell us what you set out to look at and what you found.
Well, we had Jessica and Charles Rixey in town. They originally, Charles wanted to learn how to do ONT sequencing, Oxford nanopore sequencing, so he could go to Japan and kind of replicate that work over with Kevin McKernan, who's studying a lot of these clots over there. So I offered to have him come to our lab and learn. It's much easier to do it in a friendly lab than flying to London to get this done. And Jess was going to be in town, so I said, let's kill two birds with one stone, have you guys both come up at the same time. And as we really only set out to just sequence some vaccines, and while we were doing it, we're like, well, it's easy enough to just read everything we did in the speaker paper, just to show you guys the whole process. So let's run qubits and qPCR while we're at it. And one thing leads to another at dinner with a glass of wine. We're like, well, why don't we do one more day and study this other aspect of it that we've always kind of left partially addressed? And before you know it, 2 days later, we have data that kind of reinforces this phenomena we've been suspicious of since day one, but never really drove the point home with the right experiments. So the thesis here was that all the labs now that have studied this DNA contamination issue have one particular observation that people have been scratching their head over, which is when you measure with qPCR the amount of DNA you have, you get different answers based on which part of the plasmid you look at. So if you design qPCR to look at 100 base pair region, like in the kanamycin gene, you get one answer. And then if you do it in another region like spike, you get like 100-fold more DNA. And we've always been like, is that just primer design problems or is that real? And so we set out to answer that question and have found out that it's in fact real. And we now know the mechanism of action as to why it's happening. Whenever you do something like this and you write up a paper, you think you've discovered something novel, and then you dig through the literature and find out that now someone discovered this in 1997 and we're just discovering it. And so that was kind of the story here.
Yeah, it was 2 days of work, and this is something I like to repeat because this is something that could have been done easily by anybody to demonstrate that this hypothesis is correct. So this hypothesis that we had involved RNA DNA hybrids, hybrid formation during the production process. And just to rehash for everybody what we're talking about here, as part of this process 2 manufacturing process where they used a circular DNA called a plasmid with a spike gene inside and inserted into E. coli, did an in vitro transcription reaction to get the mRNA, and it's nucleoside modified, of course. Right. The most important thing that you have to do at the end of the process is clean up your product, which is your nucleoside modified mRNA, because this was supposed to be the coding material wrapped in the LNPs, the lipid nanoparticles. So what we did in our 2 days of work, which was really fun, by the way, was we tested whether or not the enzyme that they had used at the end of the production process, which is called DNase 1, was, well, I mean, we already knew, like Kevin just said, that it's not effective at degrading RNA DNA hybrids. But another enzyme that Kevin threw in called DNase XT is. And we saw that very clearly with the qPCR that we did. It was really wild. It came up right in front of our eyes. So the thing about this is, and the reason why it was cool to see, but not surprising, is because during that whole production process, you have your DNA strand and you have your RNA strand, which is manufactured as part of the in vitro transcription, and they're complementary.
Right.
So they stick together and they do form these hybrids. It was totally predictable that this was going to happen. And like Kevin just said, it's been published before, even by one of the manufacturers, that the DNase 1 will not degrade that. Mm-hmm. So this is precisely why we're seeing so much DNA. And Kevin will fill in the next substantial gap, which involves the detection methods that they actually use, the primers that they use to pick up which DNAs that they're actually looking for. And by they, I mean the regulators, as opposed to looking for spike. So there's so many different things that they did during the production process that was clearly, I mean, it's not ignorance. They had to have known that all of these problems were going to be pending.
2 questions really quick, Kevin, before you jump in, and I'll just throw them to both of you and we can have a free-for-all. What's the extent of the contamination? So we have DNA that's encoding the spike protein and presumably the upstream regions that are, that would lead to translation, in vitro and once it's given. So we have DNA contamination. The limit to that DNA contamination should be 10 nanograms. But so what extent and which vials did you measure?
All right. So these were vials that Kevin McKernan had sent out when he knew Charles was coming to get trained. And so they're from Japan. They weren't expired. Some of them were E-lots actually, and they were 150 to I think 480 nanograms in this study when we measured them with fluorometry. Now, qPCR measurements gave us lower numbers for the reason Jess was just describing. Depending on where you measure and how you measure, it's always going to give you a lower number. Right. And the paper kind of walks through why that is. The problem with PCR is that it needs 2 primers to give you a signal. And after you fragment this DNA, if you ever have a piece of DNA fragmented between those primers, it goes undetected. The reason that's a real problem with PCR estimations is that PCR never measures the whole plasmid. It measures a small 100 or 63 base pair region of the plasmid, and then you mathematically extrapolate what might be left as a result of how much you have from that one piece of that one region of the plasmid. So if the plasmid isn't digested evenly, that extrapolation is going to be off. Likewise, if there's any cut sites that occur between your primers, then you fail to measure that molecule, and that failure is a multiplicative error, right? Because let's say you have 100 base pair amplicon and the plasmid is 7,800 bases long, you're going to multiply that number by 78. So when you miss a molecule, you miss by 78-fold. So it really underestimates everything when you use PCR.
So we know that the underestimate, we're looking at 480 nanograms versus 10 nanograms.
Yes.
As a limit. Oh my goodness. And these were Pfizer and Moderna vials, both.
Yeah, the Moderna was more on the low end and Pfizer was more on the high end.
Okay.
And I think you can see that in there. If you go through their patent estate, Moderna is spending a lot more time paying attention to this. They wrote patents and methods to avoid using qPCR to monitor this so they could get rid of it. If you're under measuring it, you don't have any motivation to reduce the number because you pass. But if you recognize that PCR is not seeing it all, you take extra effort to get rid of it. And so I think the one thing that's glaring for the regulators is that Moderna has patents from the CEO of the company that say don't use qPCR. And then they did a PCR assay looking in the wrong part of the plasmid to monitor this. So that is really damning. The second thing that's damning is that Pfizer has a qPCR assay in their EMA documents that they use to measure spike, but they never report the quantitation from that. They just use it to claim that the spike material is there, but we dare not look at how much.
Yeah.
So they know this problem's there from the CT value they got from that spike assay. They know that's hot, and then they turn around and measure somewhere else in the plasmid to try to cheat the numbers.
That seems at a minimum cavalier.
That can gets chewed up by DNase 1. So you're always going to see less of it anyway. So it's not just that they're looking in the wrong place. Like they are going to see what they want to see, which is a low amount of DNA. And, because they use another assay to look at RNA, a high amount of RNA. But just to go back to this 10 nanogram thing, this is already a ridiculous limit, we'll call it, because it's used in the context, it was established in the context of naked DNA, which is a completely different creature than lipid nanoparticle encapsulated DNA. So these thresholds that are already being surpassed are already way too high. So this is really important for everybody to understand, and for the regulators and the people who are establishing the limits, like the EMA, to look at again. I mean, we have to redefine this. And when you consider the fact that we're not just finding any old bits of DNA, I mean, we've found SV40 enhancer promoter in the Pfizer lots. This is functional. I mean, it's used in gene therapy. It's part of a gene therapy plasmid that's used on the bench to translocate stuff to the nucleus. So, none of that is good to be introducing into a human being by transfection using LNPs. No amount is good. So, yeah, I just wanted to throw that in.
Yeah, I think what is shocking some of the regulators that we've recently shared this paper with is that they were always under the impression from fact checkers that all this DNA was under 200 bases. So we went in there and showed them fragments we're pulling out that have the entire spike region encoded in it with a mammalian promoter that will make RNA once it's in a mammalian cell. And then handed them, here's all of the literature of the papers showing spike RNA or DNA persistence in patients. All right, so something is making RNA or more spike protein, years afterwards. What is it? Why is this around years later? And there's DNA in the shots that have a mammalian promoter in front of spike that contain the entire gene.
That's super scary. Oh my goodness. Yeah, so, and it makes sense. And we know that, with long COVID and some of the research, people have been characterized as spike protein production factories. How does this stop? It's absolutely insane. And when you talk to Joe Average on the street and they're on their 12th booster, it's, you want to tear what's left of your hair out. Did they know that it was there, or did they just presume that it wasn't there, or did they just know that it could be there and they chose not to find it? I mean, can you go out on that limb?
Do you mean the SV40? Sorry.
Well, SV40 and the spike, I guess, but the Pfizer and the Moderna, pardon the expression, gene jocks, would they have known that it was there?
I think the manufacturers know. And the fact that they designed a new qPCR assay to point the regulators in the wrong place suggests that they did this very intentionally to get through the guidelines in a hurry. Now, the challenge I think the regulators have is, I think during the pandemic, they weren't in the lab. They were at home. And so they had to trust the regulators on the numbers they gave them. Very few regulators have actually gone and measured this themselves. The only regulator we know that has measured it themselves is the TGA. And when they were FOIA'd for their PCR protocols, it's all black ink with one mistake they made. They left in the fact that they were targeting the kanamycin gene, which told us that, okay, that means the sponsor, the pharmaceutical company, pointed you in the wrong place, so that you would never see this. And they did that when they had an assay at hand already validated that would see the problem if they used the right one. So I don't see any way out of this being a very intentional way to hide the ball.
Right. This seems criminal. This does not seem like a civil oopsie. This seems criminal to me. I don't necessarily, if you want to go out on that limb, but it's so anticipated. And when you look at like molecular biology 101, this sounds like gene juice to me. Remember gene juice? We used to use this type of lipid nanoparticle formulation called gene juice in order to transform mammalian cells, transfect mammalian cells in graduate school. And it sounds very, very similar. And you would get these mammalian cells that were then replication competent. They could do, they could continuously produce whatever you were trying to produce.
There's actually two reasons that this limit needs to be readdressed. Jess brought up one that's very important. This gene juice makes it 100-fold more potent, and that it can get into the cell. Like naked DNA can still get into cells. In fact, Robert Malone published some of the earliest work on that with beta-galactosidase. But once you add this gene juice to it, it's 100-fold more potent at getting the cell. The second reason that might even be a larger order of magnitude that is actually coming out of Keith Peden's work from the FDA. He's the one who wrote these guidelines for whether they should be 10 picograms or 10 nanograms. And that was all based on 10 nanograms of genomic DNA only being 1,450 copies. But in his own paper, he points out that if this were a plasmid, like a viral size genome, the numbers would need to drop to femtograms or attograms, which is like 100 million-fold lower. The reason for that is that weight is the wrong measurement here. It's molecules. And 10 nanograms of 3 billion base pair DNA is 1,500 copies. But 10 nanograms of 5,000 base pair DNA is 1 million times more copies. And so we're dealing with the wrong metric here on nanograms. It should be molecules, particularly when you're dealing with a plasmid that is replication competent inside of mammalian cells and has mammalian promoters. This isn't your average plasmid in Pfizer. It's a gene therapy vector that's designed to integrate and to actually perpetuate inside mammalian cells. And in that case, you need to rip out those promoters and you need to get the copy number down to the 1,400 level. That's really the basis of Keith Peden's work. And they're not paying attention to molarity. They're just thinking 10 nanograms is all that matters, when in biology, we don't work in those metrics. We work in molarity, not in nanograms.
Just kind of break down the implications, the physiological implications with this whole gamush that is in these shots. And in terms of what we're seeing, post-injection, December 11th, 2020, in these individuals that are presenting turbo cancers, that are presenting continuous spike protein production. So what are the implications? What can happen?
Well, a hyperinflamed state for in a word or two words. There's going to be a lot of variability between individuals depending on what they got, how many they got, where it went, which cells got affected, if they're constant spike producers, because not everybody's going to be, right? These are also things we have to look at, we have to determine. But if you're constantly producing a foreign protein as nasty as spike, and I can't stress this enough. I mean, I don't know how many articles I've written on the specific peptides that are like in the spike protein. It's amyloidogenic. I mean, this is probably explaining a lot of the clotting problems we're seeing. Right. I mean, it's so insidious that the the symptomology is just completely comprehensive, but it's all immunologically based. So it seems like the best way to describe it is that people's immune systems are becoming dysfunctional, like imbalanced, which probably has a lot to do with T regulatory cells. This is in combination with clotting, the clotting pathway getting screwed up, with the clots becoming, more dense and degradation resistant as well. If that happens in your head, that's really bad. There's just a plethora of problems going on, and my worry is that a lot of people are going to end up in a VAIDS type state because of exhaustion. Because if you're constantly making your immune system or forcing your immune system to work, you're going to get exhausted populations of cells. So, I mean, that's basically what happens when you succumb to AIDS. You don't have any fighting force left over after. I mean, that's primarily because of the cells that get infected. But having said that, it's also been shown in the literature that the CD4 positive T cells are also affected by the spike protein and by the SARS-2 virus itself. So
this is, and it's consistent with what we're hearing from the people that are treating, VAIDS, vaccine-acquired immunodeficiency syndrome and vaccine injury, long COVID vaccine injury. One of the consistent features that I hear over and over again is fatigue. People that just can't get out of bed. They can't function. They can't move. Peripheral nervous system damage, et cetera, et cetera, et cetera. What else?
It also it activates latent viruses as well. So I think that explains a whole lot. I mean, I did get exposed to SARS, and the main problem I had, I lost my sense of smell, which was really awful, but it wasn't just debilitating. But I'm one of those people that has Epstein-Barr, and I don't tend to resolve it. So every time I get something, even if it's a cold or a flu, this Epstein-Barr crap tends to get reactivated. So that happened to me, and I was like, I felt like an 80-year-old woman for 3 days. Like I couldn't move my joints, and it was terrible. So I think, based on my personal experience and also what I've read, because I've read a lot about this, that is absolutely what's happening. There's a lot of viruses that are becoming active as a result of this.
Exactly. There's so much. Yeah. You're hearing so much more about these viruses that are coming up on the horizon. Kevin, what else are we? We've got all this intervening DNA floating around and protected in lipid nanoparticles. What else could happen?
Well, everyone's attention seems to go toward the integration concerns, which is certainly a concern that we need to know more about, because there are nuclear targeting sequences in the Pfizer vaccine. This SV40 component will bring the DNA there. But I think that might be more responsible for longer term health issues, whereas the acute injury actually is more along the lines of what Jessica said, which is that when this DNA just gets into the cytosol, it's stimulatory to interferon genes. It hits a CGAS sting pathway, and that sting pathway stands for stimulation of interferon genes. And so you get this sort of cytokine storm that occurs. That happens to everybody, right? Independent of integration, you're getting these lipid nanoparticles to go everywhere in the body and fire up that pathway. And if you do that chronically, Quan has a nice paper that shows that can actually lead to oncogenesis, but I think it can lead to a lot of other things. CGAS sting is also implicated in myocarditis. So we have an informed consent problem here, where we're told it goes in the arm, it stays in the arm, and it's gone in 48 hours. And the reality is it goes everywhere. And we have papers like the Krausen paper finding it in heart tissue 30 days later, the Rolpgen paper finding it in lymph nodes 60 days later, Castriuta finding it in plasmid 28 days later. We got Gonzalez showing it that it's actually in in placenta 2 to 10 days later. Hannah shows it's in breast milk. And then the OTA paper shows it's in cerebral arteries 17 months later. This is nucleic acid of spike, which is the exact target they're failing to measure. It's a serious issue in that the DNA contamination may in fact explain this persistent expression of spike that's occurring that was against the informed consent. It was not really well measured, in fact, probably hidden, I think, by the sponsors, and none of the regulators really had an independent review in this. And when they were challenged in 2023 about this, their default reaction was to not trust the independent scientists, but was to smear them as being conspiracy theorists and misinformation purveyors, but trust the pharmaceutical industry, who are the financial benefactors of this. All right, so that, something there needs to change. Marty Macri is out there doing, I think, an interesting job in reducing the hurdle rate for getting drugs through the FDA. But I think that's actually, I'm supportive of that, only if there's a different agency that reviews when the planes crash, like we have with airplanes. The FAA does not review a crash. The National Transportation Safety Board goes and does this because they know the FAA will be biased. It's human nature to bury what happened in the crash. So if we're going to open the floodgates of the FDA with Marty Macri, he better have an answer for how you're going to yank drugs off the market when they fail, because they can't seem to do that right now.
Right. Why aren't you guys running the show? I don't get it. I mean, you roll through these publications that show the distribution of the lipid nanoparticles and the vaccine. You know exactly what's coming and going. This needs to be rewound. I mean, this is a iatrogenic catastrophe, unlike we've ever seen. I mean, do you think I'm overstating that?
No,
this is the worst drug to ever hit the market.
It is. So we need to take emergency measures. Can this be, I mean, from a medical perspective, once somebody is continuously producing spike, what do they do? I mean, or they've got other intervening regions of the DNA that have done essentially permanent damage. I mean, I see a lot of practitioners just kind of throwing up their hands.
Yeah, it's a really important point. I hope practitioners are watching this. The third author in our paper, Charles Rixey, is working with Kevin McKernan in Japan on a treatment, actually, which is, in my opinion, probably the most hopeful sounding treatment out there right now. It's kind of invasive and it's preliminary, but they're saying that they're seeing very good results in real people. It involves filtering the blood. And amazingly, they're also filtering out the autoantibodies, which is a huge problem. Like I mentioned before, we have all these little peptides in the spike protein when it's translated properly, if it's translated properly, that can lead to all sorts of autoimmune conditions anew and also reinvigorate ones that people have. I mean, way back in the day, like back in 2021, I think it was even, there was a plethora of women who were talking about their endometriosis after getting the shots, going on full bore mode, where it was kind of dormant for years. And apparently that can be very debilitating. And I was like, I wonder, so just to refresh everyone's memory, if you are mounting an immune response, a specific response against a certain epitope on a peptide or a protein, which is it's just like a lock and key mechanism of the foreign antigen in your, let's just say antibodies, because most people are familiar with that.
Right.
But that little foreign protein is a molecular mimic, or if there's a homolog in some human protein. And there's a lot of papers on this in the literature that demonstrate that this is exactly the case. Then those antibodies that you've mounted in huge amounts as part of the immunological response against the spike proteins and the little bits, it could start mounting an immune response against your own proteins if they can access it. So this is what's going on. There's no doubt in my mind. I mean, it's so to filter out these guys, they're your own molecules, but you need, they're not doing you any good. So you need
Exactly.
to pour some baking soda or a fire extinguisher on the fire.
Right.
And
You're
just,
labeled as a Trump chlorine in the veins type of person on that one, but that's all right.
I guess my big question, looking back at Pfizer, looking back at Moderna, looking at the NIAID, looking at BARDA and everything is why? Why was there such a rush to get these, whatever you want to call them, these things out, to mass inoculate, a population to the tune of what, 13 billion shots? Why would you do this? I mean, is this just too existential, or any ideas?
I think that's a Bayh-Dole issue. The NIH had royalties in these vaccines, and so that made them financially eliminate all competitors like hydroxychloroquine and IBM. So the only thing that they would study was the process or the product that would line their own pockets. And so
Right.
They have over $1 billion in royalties from Moderna and Pfizer now on these products, and they don't get anything for ivermectin and hydroxychloroquine. So that led them, I think, to create policy that was favorable to their balance sheet.
and they probably, like, I'm not defending them or anything, but
Sure.
probably out of sheer ignorance, they didn't anticipate like what a disaster was going to unfold. And it really is a disaster. I mean, every single rock, I say this every time we have an interview, every rock we turn over on the subject matter of production or etc.
Right.
There's a new problem. And it's not just us coming out with stuff. I mean, the frame shifting paper, the infamous frame shifting paper in Nature that demonstrated very clearly that a lot of the time you're not actually making the protein of interest, which is the spike protein, which might actually be a good thing. You're making foreign, potentially aberrant proteins because of the N1 methyl pseudouridine thing. Like they substituted out in all of the positions
Right.
that the uridines held. So it's like 801 substitutions effectively caused slippage when you're
Right,
translating.
frame shift,
So
exactly.
that's one of the examples that always comes to my mind when it comes to thinking about the anticipatable problems. And no one can tell me that they couldn't have easily run this thing through a basic bioinformatics tool online, like to determine if there were amyloidogenic peptides, for example, or if there were going to be molecular mimics, if there was going to be a problem with molecular mimicry. I mean, these are things you can just do from a laptop. It doesn't take any time or money.
Blast and nerds. That's what we need. Just an army of nerds with blast and just look.
But we didn't have access to anything, and it's like it's only now. It's mind-boggling. Like most of what we know that now is because of independent research and people taking a lot of risk to do the work and bring it to light. Yeah. And we have to be really persistent. I mean, Kevin and I are and many others, Wafik El-Dari and Didier Raoult, and there's a whole whack of people who are just under constant attack from the Popular Network,
God
for example.
Your OCD that you can't let this go. I mean, but it's true. When you see it, you can't unsee it. And where can they find? I mean, the thing that I admire about both of you is that you spread the word. And you let people know. So where can people read more? This is in the Journal of Independent Medicine. Where else can they read about this and also your ongoing work?
I have a Substack that tends to get this information a little bit earlier. Right. It is unfortunately difficult to pronounce and remember, but if you remember the countdown of catnip, you'll find it. It's nepentolactone. And we do try to put this stuff on preprints. We've also been etching this stuff into Bitcoin and Nostr because we need decentralized tools to keep this stuff from getting DDoS attacked. For those who don't know, Wafik's papers were DDoS attacked at OncoTarget, so they didn't reach the public for about a week.
Right.
And that's not uncommon in this field. And even that frame shifting thing that Jess brought up we published about that in 2021 because it was completely predictable from a prior paper, and that got censored. So the censorship is an issue that has to be addressed, because this word is getting difficult to get out there throughout the traditional journal system. So if you don't find us there, you can find me on X as well. I tend to put a bunch of this content up there.
Yeah, Substack as well. Unacceptable Jessica. I love that name. It still remains so perfect. I'm on Twitter as well. Kevin actually just posted a couple of videos that explain a lot about what's going on inside our paper on X, and it went quite viral. You have to watch it. It's excellent. Yeah, we're just trying to go the traditional route by getting our work peer reviewed and published. We're still, playing in their pig pen, but it's unfortunate that we have to, otherwise people won't take our work seriously. But yeah, the next time we talk, we should talk about the post peer review problem that we're having.
Yes, hub peer. Exactly.
Yes.
So, no, well, I want to thank you so much for coming on the show. I mean, we needed to get this information out. And I want to tell everybody out there, spread the word, make this go viral. I mean, it needs to go more viral than the spike protein DNA. This is cataclysmic. We need to warn people. This needs to stop. We need Jessica and Kevin to run the FDA or whatever body comes
new body
to regulate the FDA.
Yes. Yeah. Well, I want to thank CHD for helping us through this, because there are various publication costs that can be challenging for independent researchers to get through, and CHD was quite helpful in that regard. So without you guys, this probably would have remained buried.
You're very welcome. Thank you. Thank you for the opportunity of doing that. And thank you, everybody, for joining us this important episode of Doctors and Scientists. Not that I do non-important episodes, but wow, I'm blown away. My day is ruined. But hopefully, take this information. Let's be useful with it. And you guys have a great day. Have a great weekend, and spread the word.
There we go.
Okay, we're live. Can you tell us your name, please?
Christine Visser.
And you are?
Danelle Van Laar.
And your sisters?
Yes.
And this is about your husband?
Yes.
I'm just going to show everybody a picture of your husband right now so that they can see him. So this is a story about him. Okay. What was his name?
Clarence Visser.
Clarence, okay.
He went by Clay.
So my first question is, did Clay take the COVID-19 shot?
No.
Why didn't he do that?
We both felt that there was not enough proven facts.
Yeah.
And it was just putting something else into our bodies that didn't need to be put in there.
Okay, so he clearly went into hospital. So what made him go into hospital? And the first question I have for you, did he have one of those pulse readings?
Yes, that is why he went in. It was reading at 77. And he had been sick for about a week. And I got one of those pulse meters, the oxygen meters, and it was very low. So I told him, you need to go into the hospital. We need to take you.
Okay, so I just want to go back to the pulse reading thing. How did you even hear of such a thing? Was it on television? Was it from a doctor? How did you even know there was such a thing existed?
Honestly, I don't remember how I knew.
Probably just going to the doctor's office.
Right,
And they
and
put it on
just
your finger.
like seeing.
So you went and bought one?
Yes.
Okay.
Yes.
And so who told you that a reading of 77 was low?
I don't remember. I've had COVID a few times, and my memory is not.
I totally understand. It just seems an awful coincidence that the majority of these stories start with the reading.
Yes.
So when you took him into hospital, was he struggling to breathe? Was he panicking, or was he calm?
He was calm. His skin tone was beginning to get grayish, and that was another reason. And under his eyes, it was beginning to get dark. And that's when he was sick. That's what would happen. So I also had COVID at the same time. So I drove him to the hospital, pulled up, he got out. And as he's walking away, I thought, you know what? Hang on. So I called him back, and he leaned down, and I said, I love you. And he said, I love you too. I have this. So he walked into the hospital, and he sat in the waiting room for quite a long time. I was texting him, trying to find out what was going on. And he says, they won't even give me anything to drink. He says, I'm thirsty. So I told him, you go up there and you demand some water. You need to have something. So next thing I knew, they had taken him back into the emergency room, and then they had moved him up to the COVID floor. And they had him on the nasal cannula for oxygen. And the next day, I was told that he was gonna be getting remdesivir. So I called him and I said, no, absolutely not. You do not take that remdesivir. You do not take it. And the nurse says, well, it's his choice. And I'm like, who's advocating for him? He's sick. Who's advocating for him? Well, it's his choice. So he ended up taking the remdesivir for 5 days.
Well, and I think you need to interject that Clay is not confrontational.
Not at all.
He was a very passive man, very loving, very generous, very kind, but very passive and believing what he was told. He didn't want to make waves.
Absolutely. And we're dealing with the medical profession. They know what they're doing, right? And in comes our government and these bonuses these hospitals are getting. And so from there, they decided he needed to go up to ICU because he needed more oxygen. He was on the nasal cannula and the BiPAP mask for almost 3 weeks.
Were you still communicating with him at this point?
Barely. My daughter remembers one of the nurses telling her that he asked us to put his phone in the closet. No.
You wouldn't do that?
No.
This is 2021, by the way, everyone.
So I finally got to go up. Let me back up here for a minute. I was told on that first day after he was admitted that my husband was deathly ill. He's deathly ill. They told me that daily. He's deathly ill. So then treat him. Take care of him. Take care of the problem. What's the problem? Oxygen. Oxygen's a problem. So when they moved him up, they put him on that BiPAP mask for almost 3 weeks. He was on both of them. I got to go up and stand outside of his room on a Tuesday before he passed. He passed on a Sunday. And I stood outside his room, and when I came down the hallway and he saw me, his big paw went up in the air. And that was like the best feeling that I had, seeing him and knowing. And he looked good. He looked good. His face was still full. The next day, I saw him again, stood outside his room. I never got to go in his room.
They wouldn't let you.
No. But I hear other people putting on the PPE and getting to go in rooms. Why didn't they allow me? So that Thursday I tried to go up there and they wouldn't let me in. And I'm like, why? We can't have you standing around wandering around the hallways. I said, I'm not wandering the hallways. I'm standing outside my husband's room. Why can't I go in? So I turned and walked away. Because,
what did they say when you said, why can't I go in?
Because they couldn't have me wandering the hallways, being in the way.
Well, and didn't you tell her that you had been up there the last
Yes.
2 days?
Yes.
And why is today different? And
just couldn't have me wandering the hallways.
Yeah. And she says, we can't have you in the way. And she, remember, he was at the end of a corridor.
Yes.
Where
it was a corner
she wouldn't
room.
have been in anybody's way.
Right. So Sunday morning, I received a call saying that they had to intubate him. And I said, I'm coming in. I'm going in his room. I will be up there very shortly, because we didn't live that far from the hospital. I got up there, and when I turned the corner and I looked at his body, the nurse was standing on one side, the doctor was on the other side, and they're both talking in my ears, and all I can see is this shell of a man that used to be my husband. His facial features were just shrunken. They were just, he was just not him. So they gowned me in the PPE and allowed me to go in the room. And I stood there with him. And he was very agitated. He kept trying to pull his hand up, move his head and pull his hand up. And I stood there, and my daughter and my son-in-law and my son finally came up. And my son couldn't stay too long. He was very emotional. And then, so I talked to my husband, and we put on some hymns and let him listen to the hymnal songs that he has on his phone. And he kept trying to reach that hand up. And I asked him, I said, do you want that taken out? And he shook his head yes. And I said, you know you're going to die, right? And he shook his head yes. And I said, will you wait for me? And he shook his head yes. So my son-in-law called the staff in and said, take it out. He went and said, out, take it out. And he, I believe he knew what he was doing. And fortunately for me, he knew what he was doing, because he didn't have to leave that burden on me to make that call if he was not coherent enough to say, take it out. And he passed. It wasn't very long. And he passed. But it makes me wonder, why didn't they let me in that Thursday? What had happened between that Wednesday and that Thursday?
And then from Thursday to Sunday.
Mm-hmm.
And you left out the part where they pulled out the tube and he was missing teeth.
Oh, he had no teeth.
Why?
I don't know if when they were putting the tube in, but when he went in, I said, make sure that they're having him brush his teeth, or they're helping him brush his teeth, because you know oral hygiene is so important for your lungs and your heart and your your blood and your body. And
How many teeth was he missing?
All of them. He had maybe a few left on the bottom.
And you said all this stuff was on the tube as they were pulling it out.
Yes.
There was, I don't know what you'd call it.
He did try to say something to me, and what I hope it was I love you. But with his lungs, they were so full of, they burned out his lungs. They blew out his lungs with that BiPAP, and the more I hear, the more I know what they did to him.
This is why it's really important that you look at the medical records, because that will tell you what they gave him on those days when he was fine and when he wasn't. Have
Right.
someone look at them for you.
Right.
I know you have them.
I do.
You just haven't looked at them.
No.
But that will give you everything, mostly that you'll want to see, to help you understand what happened. It's disgusting.
It is. Oh, one thing I did forget to say too. When I was standing outside of his room, there was a couple doors down, a group of nurses with another intubation machine were standing outside this room. One of the nurses held her hand up in the air and said, 5. Number 5, saying it very loudly. This is number 5 today. It's the 5th person they had intubated that day. And this was at 9 o'clock in the morning.
Do you have any idea, and you probably don't, but it's worth asking, if you were on a ward of unvaccinated people?
I don't know, but I believe so.
Did your husband get any more abuse for being unvaccinated, or did either of you hear that?
Well, on his chart, didn't you say it said unvaccinated, obese,
White male.
Unvaccinated,
Obese, white male.
Mm.
That's the way he was listed. That's the way they remembered him. But what they didn't know about him, he had a family that loved him. He was a first-generation American from Dutch immigrants. He had siblings.
He was a hard worker.
Oh, man, very hard. And he could walk in a room and he could make you laugh. He could make you laugh. And he could sit there with a solemn face and just, after he made you laugh.
He was the dad joke guy before there were dad jokes.
So they've taken away the most important part of your family.
They've taken away my life. He was my life. We were together 41 years, married 35. I met him when I was 17. And he was my life. He was a mentor trainer driver for a company for 28 years. He's a valued person.
How old was he?
He was 63.
And what did they write on his death certificate?
COVID was the last thing. And then there was 2 other things, pulmonary, and the other one had to do with the heart. Medically, I'm not the type of person that contains, remembers. That would be my other sister, or this one.
Has this made you never want to take another vaccine, never want to be in hospital, all the things I'm hearing as people are sitting here? Are you done?
It's, who do you trust? Who do you trust? Has it made me bitter and angry? Yes. Yes. And people ask me, why are you so bitter? Why? Really? People have told me they didn't murder him. People have said, well, are you going to get the vaccine now? No. Why would I do that? It's not the COVID that killed my husband. It's not. It is the medical profession. It is the hospital protocols, and they all did it for that mighty dollar.
Let me just show everyone, remind everyone. Your husband. We're going to put his picture on the bus.
Perfect.
Is there anything else you want to add to his story?
You know those military reels where they surprise people coming home? I always can imagine him doing that. But then I have to remember it's not. It won't happen. And my granddaughter, she misses her Paka. She talks about him. She's 5 and she just loves him. And she'll look up in the sky and she'll say, hi, Paka. But no, that's what I need to say about him.
Okay, well, thank you so much for telling us.
Thank you. Thank you.
So we won't forget him. He'll be on the bus.
I appreciate it.
This and all CHD TV programming is made possible by generous support from viewers like you. Please consider making a contribution today to keep our programming going strong. Thank you.